So good afternoon to all. My voice is audible?
Yes, sir, your voice is audible, but the screen share is not in presentation mode. So first you have to keep the slide in presentation mode.
Okay, 1 minute.
Now it is visible, sir, but keep it in presentation mode. Yeah. Now okay, sir.
Okay. Thank you, sir. So almost it's late. So anyway, I will finish my presentation as soon as possible. So first, now I'm going to explain about the mass production technique of insect predator. I think the introduction is not required. Everyone knows about the biological control. What are the biological control? So insect predator, parasitoid, pathogens, everything maybe they know for introduction. So directly I skip some slide. Directly I'm going for predators only, direct predators. So this also everyone knows what is a predator, what is a purpose and things. So I think these are very basic things. Out of 30 orders, almost 16 orders have insect predators. More than 200 families, more than 2 lakh species of arthropods have predators. So you know that is some insects have monophagy, oligophagy, and polyphagy. These are some basic characters of insects. So just I will make it some slides for what are the important predators that is there in our field. So many are there. For example, praying mantis are there, dragonfly, damselfly, etc. But it's very difficult for mass produce of all the insects. So few things only it is possible. So just I will explain. Earwings, everyone knows. These are very important predators. Ladybird beetles like coccinellids, the redweed bug and pendadomid bug, and this anthogorid predatory bug, big-eyed bug, and lacewing. These all things is very easy for it is very common predator, and also some insect is possible for mass protection. Now directly I'm coming to the point for mass protection technique, because I think this introduction part is not required. So I omit some slides. So here, the first slide, I am going to explain about the redweed predator. You know that redweeding under the hemipteran insects, almost 99% of the redweed bugs are predators only. Few insects only, you know that some insects for hematophagous insects, it is spreading the Stocker's disease. You know that's a blood-feeding insects under the subfamily Tritominae. That's only one subfamily only there. And remaining 99% of the redweeds are insect predators only. One more insect is there like Ectrichodinae. There's a subfamily Ectrichodinae. There's a millipede predator. It's feed on only millipedes only. So here you see the, now I'm going to explain only the focus on insect predators only. Here, what is the problem is there in the field? The field identification is very, very difficult for identification of the pest and predators. For taxonomical, so many characters are there. But you see here the mouth parts, they're very lean and long. This is a pest actually, red cotton bug. This is a pest. This is a predator. The first identification is the mouth parts. The mouth parts is very short and stout in the predator. Here is a predator. At the same time, here the pest, you see the mouth parts is very lean and long. This is one of the major identification. Whenever you go to the field, you see the mouth parts, then it will come for conclusion. The second, you will go for the some small experiment. You know that insects experiment, all the things. So basically, to identify the insect, pest, and predator, then you will go for the identification of the insects. Particularly for this redweed predator, maximum is preferred. You know, this is a hemipteran insect, so piercing and the second type of mouth parts is maximum to prefer the lepidopteran caterpillars. Basically, these three species is very common: Rhinocoris marginatus, Rhinocoris fucipus, and Cycanus colaris. These three species is very common in all ecosystem, including pulses, cereals, and vegetables. This life cycle, you see the life cycle total. You know that the hemipteran insect total 5 nymphal instar, total 45 to 50 days almost it is there. Then it will come for the adult. The fecundity, you see, is a very high fecundity. It's almost minimum 1,000 to maximum 2,000 are there approximately, the fecundity. So this is a different stage of insects. 5 nymphal instar, then it will come the adult. Here both nymphal and adult, both are is predator only. So now I'm coming to the point for mass protection. So it's very easy method at farmer level. That's why because this training related to the organic farming. So whatever I'm going to explain here is related to farmer level only. It's very easy method, no need of any hi-fi technology. Instrument also nothing is required. Just you will take one plastic container, plastic or whatever container, no problem. 2 centimeters sand or soil, just you keep the soil. Just to spread the water and sunset for 2-3 days. Nothing scientifically will go for the sterilization of the soil. That's all. So you will take big container or small container. There is no problem. The next important point, you will put some corrugated paper. Corrugated paper means nothing. Just you will take chart paper, but you will take black or brown color chart paper. Why you prefer the black color means maximum all insect. I think everyone knows. Maximum that insect is prefer the dark place only for laying the eggs. So better you will take brown color or black color chart paper. Just you will make it a zigzag. Okay. So you prefer the zigzag the chart paper. Just you keep inside the chamber. The next important point is host. You know the host. Maximum you prefer the natural host like Helicophorus, Podoptera, or any other Lepidoptera and Scatterplus, no problem. But larva, that is very, very important. Otherwise, laboratory host, better you will take for Carcera cephalonica. I think that's a very easy method for mass produce of this Carcera. So what you will do, nothing, you will take any grains like sorghum grains or bajra, whatever it is there. Just you will add a little bit for groundnut, okay, for protein. Just you will mix, for example, one big container you will take like the same plastic tubs. Then you will add 2 to 2.5 each of grains and mix it with the groundnut. And you will add a Carcera egg. You collect from any laboratory, biocontrol laboratory. Carcera egg is very common. Just you will take, they are given the cc, 1 cc. 1 cc means approximately 16,000 to 18,000 eggs. Okay. Just you spread off cc is enough for one tray. Then you spread the egg, you cover with muslin cloth. Just you will keep for 20 to 25 days, 25 to 30 days. So what happened? That larva, it will come from the trays. Then for further mass protection, same thing you will use for, you collect the adult, then you keep with oviposition chamber, then you collect the eggs for further mass protection. Here you collect the larva for mass production of this readable. We are using as a host. This Carcera is a host. So this larva, you will take the larva, then you release inside the chamber here. For example, small container, for example, this container size almost 30 centimeter length. So you will put more than 10 pair of adult, no problem. Small container means you will take only one pair. One pair means one male, one female. You know the insects, the females are dominant than male. The size of the abdomen also bigger than male. The female is size is very big. So you collect both male and female. Small container only one pair is enough. Bada size means you will take more than 5 or 6 pair based on the container only. But whatever insect you are releasing here, the same host also very important. For example, only one pair. One pair means 2 insects. So we'll add 4 larvae is enough. But you're releasing 10 larvae, 10 pair means you'll put 20 to 30 larvae. So selection of larvae is very, very important. Because if any contamination is there, for example, any fungal infection or viral infection, something it is there, the larva, maximum this predator is not prefer that type of the insects. So what is required? Healthy larva is very, very important. The second thing is size of the larva. The size of carcera means you will take maximum 5th instar only, 4th and 5th instar larva. The very big size of the larva. Spodoptera means you will take 4th instar. Don't put the chota larva or bada size larva. Okay, that is very, very important. Selection of larva is important. The healthy of larva also is very, very important. So after releasing the host and nucleus culture of this predator, just you cover with muslin cloth. Every 2 days once or alternate, whatever it is there, just daily also no problem. Just you will check inside the chamber. The female maxims lay the eggs on the chart only. The black color chart is there, na? That is exact method. Maximum the female is lay the eggs in the group. You see the photo here is almost is in cluster form. So one cluster means it is maximum 45 to 50 eggs are there minimum. So maximum 60 also did that. So just you will cut the paper, you keep in the petri plate. How you are making the petri plate, you see here. So here one important points. For example, summer, for you know that insect rearing temperature is almost 30 plus or 2 only temperature. Humidity is required 65 to 70. Suppose the temperature is very high, the humidity is very less, means just you will keep some wet cotton inside the petri plate for catching the eggs. The incubation period means it's a catching duration. Maybe it is there for 7 to 10 days. So after within 7 to 10 days, after 1 week, you see the eggs, the nymphs it will catch. It's very small, look like ants only. It's very small size. Okay, this is I took only one photo. This is a new first nipple instar. This is very important points here. From first nipple instar onwards is a predator only. So what is required next is required for larvae. So after catching, immediately you will add some larva in small size of the larva. For this is a first instar, na? So you will add first or second instar of carcera larva. That is very, very important. So don't add for a big size and medium size of larva. It's a very small size of the larva is enough. For example, one petri plate means it will come for 40 to 50 nymphs. Correct, na? 40 to 50 nymphs is there. You will add 1 or 2 larvae is enough. Don't add the 40-50 larvae inside the petri plate, because you know that carcera they're making the webs inside the petri plate. To avoid the webs, what we will do? We will add 1 or 2 larvae, small size of the larvae, because the first feeding is very, very important. So after that, there is no problem. So you grow only first and second instar only under the petri plate. So for example, just you will grow 1 week the same petri plate. After that, you will make it for 2 petri plate, the second instar. So up to the second instar, you will add a limited number of the prey only. Just you will add 2-3 larvae, maximum 4 larvae is enough. Small size of the larva. The first 2 instar means almost 10 days only. 5 plus 5, 10 days only. So after the second instar, this coming the third instar. Third instar means after second molting. So that time onwards is required more number of larvae. So that's why I'm always recommending for third instar is a correct time for field release. Some people I saw there, so many people they are growing lakh number of the predator. Simply they are releasing the adult stage. It's not correct. Adult, you know, adult have wings is flying. So it's a better compared to the adult. So the third nipple instar is a correct time for field release. Okay. So better you will take third. This is third nipple instar. So that nipple period 45 to 50 days. The first 10 days you will grow at laboratory condition. Then after that, directly to release into the field. So whatever it is required for further mass production will grow at a laboratory condition. Otherwise, whatever it is that directly release into the field. So this is third NIPHM instar. This is a fifth NIPHM instar. This is adult. So this is a life cycle and mass production technique. So here the adult is almost living for a minimum 6 months. So it display the one single female itself only display the eggs almost more than 2000 eggs. Just you imagine, fecundity is very high. So no need of grow is lack under lack number. Just will grow is less number of the predator. The whatever nymphs is coming directly released into the field. So the fecundity, that is a feeding potential. And the field release as per the recommendation is the 3000 to 5000 per hectare for larvae. You see the video here, just I took the video from our NIPHM campus, how that predator is feeding the larvae. I think this is a spot after fridge bada in paddy, in a maize ecosystem, maize field. So the next predator is a pentadomid predatory bug, Pendadomidae. For example, Echinocona facilita is there and Antralysis. This too is a very good, very common predator in our field. For example, this paddy or maize or whatever it is there. These two species under the Pendadomidae is very common, including for vegetables also. So here, compared to the redwood bug, this is very short life cycle only. It's a very good predator. So you see here, this is very important points. I'm telling, just I explain some bullet point only. I think these all the things is not required. Here, what happened after egg laying, the catching after catching, the here the incubation period is very short time, 3 to 5 days only. But here, after catching, the first instar is there, first instar. The first instar is act as a pest only. It's a very, very important point. So after catching, the first instar is required some fresh leaves. So better you will take brinjal leaves, fresh leaves. Just you'll keep inside the container after catching. After catching, first feeding is required some fresh leaves. Brinjal leaves is good actually, because I tried so many things. Brinjal is it is good. Brinjal leaves, it is success for 100%. There is no mortality. If other leaves means some mortality there. To avoid the first instar mortality, we'll keep some fresh brinjal leaves. The after first feeding, no need of any leaves. So the second feeding onwards is required some larvae because it's a predator. So I'm telling the repeat. First feeding, not first instar. First feeding only it is required leaves, fresh leaves, brinjal leaves. Any other leaves, no problem, but the brinjal leaves I get good result. The second feeding onwards is required larvae. So what here also same procedure, you will add small instar, small larvae only. Live larva, that is very, very important, and healthy larva, that is also very important. For example, 50 nymphs are there. For example, I'm telling 40 to 50 nymphs are there. You will add 4 or 5 larvae is more than enough. Then the second instar will add, increase the number. Third, you will increase the number. Okay, based on the things you will add. Here also total 5 nymphal instar. Then finally it is coming the adult. Here also both nymphs and adult, both are a predator only. Is feeding the larvae. The third important predator is an anthoporid predatory bug. It's a very good predator against thrips and mites. So this insect mass protection technology derived from NBIR director, retired director Dr. Sandesh Pallal madam, I think everyone knows. It's a very good predator actually. So I will play this video. I think it is feed. Yeah. You see here this photo I took from our NIPHM campus only. You see here, this predator is feed the first instar of Falamivo larvae. So this is the adult for Plastidis species. So how is feeding? Yeah, here I'm telling this important points. For mass protection, it is required French pin. You see the eggs, how the female it is lay the eggs in the French pin only. This is a eggs. Is not lay the eggs like other predator. It's required the French pin. Inside the French pin only, that membrane, that French pin only. This maximum lay the eggs there only. So this is a eggs of Plastidis species. This is the nymphs. How is feeding? Is feeding on the mites also, because this is a very good predator against mites and thrips particularly. This predator is fed on the eggs of Spodoptera frigibada, including eggs also. You see the life cycle. Biology of these insects. The incubation period maximum 3 to 5 days. The total nymphal period 5 nymphal instar. Minimum 14 to 16 days. The total development period is almost 70 to 23 days. Here, fecundity is very difficult for seeing that eggs. That's why we are calculating for nymphs for female. It's almost 110 to 203. It's very huge number is there. It's very easy for mass protection of these insects. Yes, its fecundity is very high. Now I am coming to the for mass protection. Just you will take beaker. It's almost a 2 liter of beaker, glass beaker. Then you will add one tissue paper, first layer tissue paper. Then you'll put some cotton, absorbent cotton. Okay. The next important points, food is required. So here, what is the in laboratory condition? You will add Carcera eggs only. So you will put the fresh Carcera egg, means it will catch within 3 to 5 days. To avoid the catching, what we will do? Better you will go for UV treated Carcera egg to sterile the Carcera egg. So UV treated is very, very important. After UV treated this Carcera eggs, just you spread inside the beaker. For example, one beaker means you will add just not half cc also, just 1/4 of cc is enough, more than enough. Just you will add a little bit, small quantities enough. Just you spread the Carcera eggs or above the cotton. Then you will add nucleus culture of this Andhra predatory bug, Plastidis species. So just here for a maintain of humidity, just you will add a little bit of wet cotton also, just to stick side of the wall. The next important points, after this adding all the things, you will add, already I told, is required French pin. Just 2-3 pieces enough. So just you will make the piece of French pin and you keep inside the container. So what happened? This female is lay the eggs in the French pin only. Don't try any other vegetables. Already we have tried all the things. Maximum prefer only French pin only. I don't know what is the reason, what is the things. I don't want to explain about all the technical things here. So better you will take French pin only. So this female is lay the eggs on the French pin. What you will do daily or alternate day? Just you collect the this French pin. Then you will add the Carcera eggs for feeding, for food. The second you will add French pin, fresh French pin. Okay. So just you collect all the French pin, you keep in the separate chamber. Just wait for 3 to 5 days. the nymphs will catch. Then after that, you see here is, for example, for 5 small French pin means you will take almost more than 100 number of the nymphs will come. You see here, this dot is there, small red color dot. These all are nymphs of blastocysts only. Here both nymphs and adult both are predators only. So whatever the further mass production, you go for the same method. Otherwise, directly you release into the field. Just you take along with this cotton, then directly release into the field. You know that compare to open field in protected cultivation in polyhouse condition, greenhouse condition, how farmer they're facing the problem for mites and thrips. For this mites and thrips, this predator is a very good predator for control of thrips and mites for management. So here I'm telling some important points. You know that predator means cannibalism is very common. To avoid the cannibalism, is sufficient quantity of food is very, very important. So we will add UV treated carceri eggs. That is very important points. Not only for this predator, all insects same like that only. The second important points, so French pin. Okay. So whenever you will add the French pin, just you will check if any fungal or any contamination is there, just you avoid the type of things. Otherwise, you will change the ovipositional chamber immediately. So cleaning is very important. The next important predator, green lacewing. Everything we know this predator is very good predator against aphid actually. That's why we are calling aphid line. This that's why we are calling for aphid line. So under the Neuroptera, these insects. Yeah. It's a neuropteran insects. You know that all neuropterans are predator only. Common name is green lacewing or aphid line. Okay. So you will go for the publication. Here they are mentioned for all sucking pests. They are mentioned for thrips, mice, dacids, everything. But frankly, I'm telling this insect, this predator masses prefer for aphids only. Suppose aphid population it is not there, it will maybe is feeding off other insects also. So this is a life cycle of these insects. So this adult. The adult is living almost minimum 20 days, is maximum 60 days also there. So the female is lay the eggs for a stacking egg. You see here, the photos look like white only, but its original is a light green color actually. Okay. One single female itself only display the egg almost 300 to 500 eggs. Some publication is there 800 also. Fecundity is very high fecundity. Then the eggs will catch within 3 to 5 days incubation period. Then that's a grub stage. This grub stage only is a predator. Adult is not predator. Grub stage only is a predator. The grub stage almost 10 to 12 days only. Then it is coming the pupa, then pupa is emerged into the adult. You see the grub stage. This Chrysoperla is feeding the eggs of Lepidopterans. Maybe this Frugibada only, I think. Yeah, Frugibada. Frugibada egg. Follow me. I think it's the third stage. Yes, we'll omit this one. Okay. Yeah, next I'm coming to the point for mass protection of this Chrysoperla. So if you want, you will take this beaker types, same 2 liter beaker. Otherwise, in online, this is available for this Chrysoperla breeding nest. You know, India Mart also is available. So you will take the oviposition chamber. You will put one tissue paper, one layer. Here, already I told, Chrysoperla adult is not predator. So it is required in natural condition. What is they prepare from the nectar or pollen from the flowers. But in laboratory condition, you will prepare the artificial tide. Very easy method. Just you will take yeast, fructose, honey, proteinase, and water. These 5 content you will take 1:1 ratio. Just you will take pinch enough, very less quantities enough. Each and every day you prepare the fresh tide. That is very, very important. So whatever I mentioned here, just you will take 1:1 ratio. Just you mix, soak with cotton, and keep stick in the side of the wall. That is very, very important, because some people they put that along with more water or more honey. So it is staying here. Better to avoid this one. Just spice all the things, unwanted things. Just a little quantity is enough. Okay. Then next you release the adult. Here no need of any host, because the adult is not predator, is required for this artificial tide is enough. Nucleus culture you take from any laboratory, then you release inside the chamber. Maximum they are given the people stage only. So both male and female, you will approximately for 2 liter beaker means you will release 10 pair is more than enough. So this covering is very, very important here. Previously we are covered with muslin cloth, but here you will cover with brown or black color chart paper only. Just you will make it the lid, and also you will make it the small holes with help of pin. Just you will make it the whole center part of the chart paper with the help of rubber band. Just you will cover this beaker. Maximum this female is lay the eggs on the brown color paper, brown or black color chart paper. So you see that already I saw that egg mass, how the eggs are there. So it will catch within 3 to 5 days. This is the first stage of grub. Here grub stages are predator. So very common, cannibalism is very common. To avoid the cannibalism, what we'll do, each and every, for further mass protection I'm telling. So what we'll do, each and every grub should be maintain the individual container. Better you will take syringe bottle. Just you will add a UV treated carcer, because a predator, so it's required carcer eggs. So UV treated carcer eggs only, just you will add a small quantity each and every container. Then directly you release each and every container is one predator only 1 grubs. That is enough. So just will grow maximum 10 to 12 days, because the 3 grub stage. So after it is coming the people stage, so that pupa just you release into the oviposition chamber for further mass production. So this is a life cycle and for mass production technique. For field release, here that is very, very important for field release. The grubs only predator. So what we'll do, you directly release in the first instar grub stage. That is very, very important. Otherwise, what we'll do, the eggs are there, na? Just you collect all the eggs. Just you will add a little bit quantity of trichogramma, you mix and directly release into the field. That option also there. Otherwise, we will make it for like how we are releasing the trichocard. Na? Same method, you will make it for chrysocard. We will stable it in the field wherever the aphid population is there. Na? Directly release into the field. That option also there. But this is a very good predator, aphid. Aphid predator. So field release almost 50,000 per hectare during the season with a cap of 15 days interval. The next important thing is ladybird beetle. Everyone knows that coccinellid is one of the good predator for aphids and also other sucking pests. So these all are common points that I escape something. Yeah, here. So this is a egg mass. This almost 1 egg mass means 8 to 12 eggs are there. Here also 3 grub stage. Then it is coming the pre-pupa, pupal, then adult. Here both grub stage and adult, both are predators. Here it is required. Already I told this is a aphid feeder. So host is required for aphid only here. It is very difficult here with the Coursera. It is impossible. So it's required aphid. So how we will grow the aphids? It's very easy method. You will grow for mustard because mustard seed is each and every kitchen is available. You take one small container, just you fill with trichogramma and vermicompost, and you will spread the mustard seed. It will grow. Just you will maintain a 10 to 20 trays. That is very, very important. After mustard growth, the maximum naturally it will attract the mustard aphid. Otherwise, you release some nucleus culture inside the mustard. It will grow within 20 to 30 days. So now aphids are ready. The aphid is spreading the mustards. Then you will take only one container. You release 10 pair of ladybird petal. Here I'm telling one important points. Maximum you prefer the local species. Whatever the species available in your field, you select only adult stage. That is very, very important. Don't prefer the grub stage, because grubs are maybe it is there some parasites by some parasitoids and some other predators. To avoid that one, you prefer only the adult stage. So just you release inside the chamber whatever the aphid infected plant, just you release. Here some points is there, because you know coccinellids have more cannibalism compared to other insects. To avoid the cannibalism, what we'll do, you make it the cylinder, small cylinder type of thing. Just you will take brown or black color chart paper, you will make it small holes like that. You make it. You keep inside the container here and there. Almost one chamber means you keep over 10 holes like that. You see almost here a 2 to 3 centimeter length. Okay, the width almost 0.5 centimeter only, very small only. So what happened here? This female maxims lay the eggs. Maxims prefer this type of because the predator, what is the character is there is need dark place. So if you want, you will go to the field. If you want predator, wherever that field in a shrub area, that is corner side, if any shrubs are there, any stones or something be there, just you will open. You will open below the plant. Maximum we will collect the predator. That is a character of predator actually. So what we'll do, just each and every day you will check that this chart, the chart hole. If any egg mass is there inside, just you will keep inside a separate petri plate. So within 3 to 5 days it will catch. So after catching, immediately you will release fresh aphid infected plant. Don't mix grub stage and adult. Don't mix for a different stage of grubs also. For example, today I have 50 eggs. 50 eggs are catch. For example, 15 grubs are there first instant. So you collect all the grubs. You release separate container only. Infected aphid and aphid container only. Don't mix for second insert, first insert, third insert, and first insert, make adult and the grubs. Each and everything you should be maintained the separate chamber. So here one more problem is there for people stage. For example, after 20 days, 20 to 25 days, it will become the pipa. So pipa, what we'll do, just egg case is there, na? If you see the photo. So this egg case you keep inside the chamber. So automatically that pipa, it will go for pipa, na? The last stage, the all grub stage is moving towards the here only. That's just naturally it is moving. So each and every egg case you have that people stage, because to avoid the people cannibalism also. Because what is a problem is there here? Which one is emerging the first adult? This adult it will disturb for this pipa cannibalism is there. So this is a cannibalism video. What in this one? No need, I think. Yeah. So here, so whatever after catching directly release into the field, that's the best things for the mass production. You will go for the same method, whatever I explained here. So a lot of publication is there for artificial data. Frankly, I'm telling, I also tried here. it will grow for first generation, but the after generation we are facing the problem for artificial diet. The fecundity is very poor. So better, as per my experience, better will go for the natural diet. The next thing is not insect anyway. I explained this also. Spider is one of the good predator. So it's very easy for product mass production, but the problem for here cannibalism is very common. Is compared to the other insects. Here is a more cannibalism is there. You know that more than 39,000 species of spiders from 1 ton families. So I simply avoiding some slide. Yeah, here you see there's a very important points that here what is the beauty of the spider? Spider is lay the eggs. It's like ovisac. Yeah, come. This one. So ovisac, one ovisac means is almost 200 to 300 eggs are there inside the ovisac. Yes, here only finish. So almost per female is lay the eggs almost 2 to 3 ovisac only during the life cycle. Okay. This nymphs we are calling for spider link actually. Spider link. The spider link almost 6 to 8 months. So here no need of for rearing all the stages. Only you rear the adult stage only. Now I'll explain here. It's very easy method. Just you will take one big container. You see the length of the container is almost 30 centimeter length. The width only that 10 to 20 centimeter only. 10 to 15 centimeter only. The second important points. So it is you will take one syringe bottle. You keep some fresh leaves, but you will take ornamental plants like a golden turanda, whatever it is there. Just you will pour the water here, then you will add this leaves. So whatever prepare that leaves, just you will keep inside the container. You will make it the natural condition inside the container, oviposition chamber. That's all, nothing is there. Okay, just you will keep inside the oviposition chamber. So next is a food. So better you will take for artificial this laboratory host like a carceral larva. Otherwise, any host, that's no problem. So just you release inside the oviposition chamber. Then next stage is you release the adult spider nucleus culture. Better you will take both male and female together, no problem. But you will keep only 1 week only. After mating, you remove the males. Only female is enough. So here every week 1 or 2 times is enough, no need of daily. So alternate day, just you release the host inside the oviposition chamber. Carceral larva is enough, 1 or 2 larva is enough. So here this female is lay the eggs on the where in a cloth or the plant only. This is for a ovisac. So here what is the character is there is always the spider female is attaching with that ovisac only. It's almost within 20 days the egg will catch. You see the one ovisac means 200 to 300 eggs are there. So after 20 days, the spider links, you see these small things, spider links. So almost 200 to 300 spider links is coming from the single ovisac. Here for further mass production, what is the problem is there? So each and every spider links is required 1 chamber. It is very difficult. So as per my experience, better after catching, for example, 300 spider links is there, directly release into the field. That is a good to increase the spider population in our field. So better what we will do now, whatever the spider adult is there, just we collect from the field, you will rear, because in natural condition a lot of problem is there. So to conserve this predator, to think about that way, so you collect whatever adult is there. Maximum the field condition means already is mating. So you collect the female, then you rear in the laboratory. After ovisac, so whatever the nymphs is coming, directly release into the field. So yeah, this is for mass production. The last predator, this one everyone knows about this predator, is a Cryptolemus monstrosity. It's a very good predator for mealybug actually, and scales also. All the things on body. Yeah, here. That's why we are calling for mealybug testaya. Here both grub stage and adult, both are as a predator. So this is a adult, is a eggs. If for female is almost is lay the eggs 200 to 300 eggs. These are 3 stages, grub stage. The fourth instar is a pre-pupal stage, then pupa, then comes the adult. For identification, both male and females, the size of the insect almost compared to the males is bigger than females. And you see the foreleg. The foreleg, the brown color, light brown color means it's a male actually. The dark black color means it's a female. This is one of the major identification of male and female. And directly coming to the point for mass production. So better you will go for this pumpkin method. You will take pumpkin. No problem. 130 up to 130. So you will take the pumpkin. Just you prefer the pumpkin is very important. So healthy pumpkin is very important, and without any damage with the stock. So you will keep some. You select the pumpkin, and you will add the nucleus culture of mealybug. Because the host is mealybug. The prefer of the mealybug is very important. So better you will take from a laboratory. You will prepare the species Macrococcus quercetum, that's we called for pink mealybug or crepes mealybug. That species you will take. Just you will keep small quantity of nucleus culture in above the pumpkin nearby star. So it will spread within 20 to 25 days. The whole pumpkin is infected by this mealybug, Macrococcus quercetum. So after you see the rack how it is there, the whole pumpkins are infected by mealybug. Now mealybug culture is ready. So next we will go for the predator. You select the predator from any laboratory. Nucleus culture you will take. Approximately about 10 pair is enough. So just you release the infected pumpkin mealybug. Sorry, infected this pumpkin. So whatever the mealybug is there. So here, what is the character of this Cryptolemus? So wherever the mealybug it is there, this female is lay the eggs there only. Okay, so culture is very, very important. Mealybug culture is very important. The female is lay the eggs along with mealybug colony. So within 4 or 5 days is enough, or 3 days more than enough. After that, just you collect all the adult, Cryptolemus adult, then you release the next chamber. Okay, next infected mealybug, wherever that pumpkin is infected, just you release there only. So the eggs will catch maximum within 20 days, 20 to 25 days, the whole pumpkin, wherever the mealybug infected. So all they will come, you see the here, the mealybug, Cryptolemus, they will spread. So that now there is no mealybug. So all whatever you are seeing, this is a Cryptolemus only. The Cryptolemus grub stage is look like mealybug only. So one pumpkin means almost 300 to 400 Cryptolemus adult will emerge from the single pumpkin. Sorry, some video. Yeah, no problem. I am avoiding this video. So what is the recommendation? Is 2000 per acre or 10 per plant or 20 to 30 per tree. You know that this species mealybug, you know, what are the crops, how is infected, everything is there. So whatever I explained for this all predator. So conservation is very, very important. And the second important point to avoid the insecticide spraying. So battery will grow for some ecology engineering plants around the buns for nectar and pollen and shelter, and also alternate prey for built up our natural enemies. That is very, very important. So whatever we are rearing and releasing the field, that is one of the things. And also conservation is very important, and to protect our biodiversity. So thank you each and everyone for listening my class. I explain only the predator. So if any clarification, I will clarify now only. Otherwise, I hand over to Dr. Sailaja. She's waiting here only. So she's explained about the parasitoid. Some small section only. Just she's going to cover within 20 minutes for parasitoids. Then after that, we will go for the clarification.
Any participants have any doubts?
Yeah, sir, why predators oviposition happens in corrugated, sir? Why they are folded? What is the reason behind?
Yes, sir. Is nothing, sir, because maximum predator is prefer the dark place actually. Dark place or it is that is a natural thing, sir. Masses prefer the dark place. That's why we are putting the brown or black color chart paper. Not only for predators, sir, all insects, including Spodoptera or whatever it is there. I think if any entomology is there, they're using in biocontrol lab, they're using for brown and black color chart paper. Because of oviposition only there, nothing. It's prefer the dark place.
No, does it have any relation with the ovipositor? Is convenient to lay eggs in the foldings?
No, sir, it's not like that actually.
Can't they lay eggs on the plain paper, sir?
But sir, what sir?
Can't they lay eggs on plain paper?
Is compared to, yeah, it is there, it is there. Compared to the plain, just you will keep some corrugated. Is for we are checking both also, but compared to the plain things, we get more eggs of this place only. There's a lot of publication is there. Just you go through that publications.
Sir, I have lost your voice. I can't hear you.
That number is a difference. Hello, sir. Is this clear, sir?
Yeah.
Yeah. Sir, is compared to the open things. So what are the we are using that dark place things, sir, like a chart paper, that zigzag method. I am explaining.
Yes. Yes.
So compared to the open, this one will get more fecundity. We are collecting more number of eggs.
In field conditions, I observed any dark cloth, like a shirt cloth or any material, fabric, if they are placed on nearby the fields, we see plenty of stocked eggs by Chrysoperla, sir.
Yes. Okay.
This
Yes,
can
sir.
be this can be a good breeding ground for
Yes.
these predators. So
Yes.
I recommend all the farmers to just flag few dark cloths in
Yes.
and around their fields.
Yes, sir. Correct, sir. Good, sir. Yes, sir. Yes. That's why we are using in a laboratory condition also. That is one of the reason. But I don't know scientifically what is the things. Maximum this predator is prefer the dark under that dark place under that shadow place. Yes, sir. It is there.
Any other participants? Okay. Thank you, Dr. Jesu Rajan, sir.
Yes, now I hand over to Dr. Sailaja, sir. She is going to explain just 20 minutes only.
Okay.
She's covering the parasitoid also, sir.
Okay. Sure, sir.
So now I hand over to Dr. B. Sailaja. She's working as an assistant scientific officer in entomology. Now she's going to explain about the mass production of important parasitoids.
So good afternoon, everyone. So is my voice audible?
Yes, ma'am, very much.
Okay. So just I will brief about important parasitoids that can be mass produced very easily at farmers level and at laboratory level. So in that many of us, we know that what is the parasitoid and different types of parasitoids available. So parasitoids are the insects that will use the host insects stage, either it is egg stage or a larval stage or pupal stage, for completing its life cycle. So meanwhile, the internal body content will be used for its development and the host will be killed. And these are the different types of parasitoids types I mentioned. So in this site of oviposition and number of parasitoids developed from the parasitized host and a stage of parasitization. Based on these parameters, these different types are all there. So I'll skip this one. I have mentioned only the examples, and where I will show the mass production, I will explain about this. So here I have given the photos of Trichogramma. This is the first one is a Trichogramma. This is a egg parasitoid, and this is a Gonios Nephron, very much useful management of larvae of black-headed caterpillar. So for a year-round production of whether it is parasitoids or predators, we need to have the good quality and quantity of host. So for that purpose, we maintain the cultures of this Coursera cephalonica, that is a rice moth that can be easily multiplied at the laboratory level. So here for mass production of these, we need to have any unused containers like plastic buckets, plastic tubs we can use. So in that one, we need to take the feeding material. So depending on the availability of the grain and cost, so we can select any millets and cereals for growing of this Coursera. So for that, we need to sterilize the grain to reduce the moisture levels because we need to wait for 45 to 90 days for adults emergence and for collection of adults. So meanwhile, we may face the problems of fungus attacks. So for that purpose, we need to reduce the moisture by drying the grains under the sun or in the oven at 100 degrees centigrade for 30 minutes. So after that, we will add the eggs in that. We will take the feeding material and we will sprinkle the eggs, but after sprinkling the eggs, we should not touch the grains because the eggs may get damaged. So after that, we will tie it with the rubber band, we will put a muslin cloth and we will keep it in a racks untouched for 35 to 40 days. So after that, every day we need to observe the tubs so that we will be observing the adult emergence from 35 days onwards. So at that time, when we observe the adult emergence, we need to collect the adults and we need to collect the eggs laid by the adult insects. So freshly laid eggs will be collected and that will be strained and used for the mass production of predators and parasitoid. So very important, very popular one of parasitoids are Trichogramma group. This is a egg parasitoid. In laboratory, we will be mass producing the Trichogramma species in Coursera eggs. So one important thing need to be noted while mass production of Trichogramma is that as it is a egg parasitoid, at laboratory level or farm level, we should not get the host larva. So for that purpose, we need to sterilize we need laboratory, we will take the Coursera egg and we will clean the Coursera eggs and we will be pasted on a thick paper chart with the measured quantity of Coursera egg. After preparing the Coursera egg card, we will sterilize the cards under the UV light, and the card we need to write that what is the date we have given for the parasitation and what is the species we are giving for parasitation. That things we need to note on the paper. So we will be keeping for 30 to 40 minutes for killing the embryo of Coursera egg. Later that egg card we will remove and we will use for the parasitation. So what we will do, we can use any glass tubes or a plastic covers for mass production of Trichogramma. So first we need to have the Trichogramma mother culture. So that Trichogramma mother culture we can procure either from the NBIR or from NIPHM. So the tube which is having the mother culture of Trichogramma, in that we need to put the Coursera card, that is UV sterilized Coursera card. So after that, the females of Trichogramma will parasitize the eggs of Coursera, and after 24 hours, we need to remove the Coursera parasitized card. That is a tricho card. That tricho card we need to remove and we need to give the fresh card for parasitation. So like this we can do until the adult Trichogramma die. So when we remove the card from the tube, after 3 days of parasitation, the egg color will change to black color, means that is a parasitized egg. So here you can see the life cycle of Trichogramma. This is a female of Trichogramma inserting egg into the host egg. So after 1 day of egg period, the immature stage, that larvae of Trichogramma will develop, and after 3 to 4 days, the parasitized host egg will turn to black color, and that contains the pupae of this Trichogramma. So if we, this is a stage where we need to release in the field, or we need to store it in the refrigerator up to 15 days. So if we keep the cards in open condition up to 4 days, means the adult of this Trichogramma will come out of the parasitized cards. So parasitized, as I told you, parasitized cards we can store up to 15 days. But if we store more than 15 days, means the emergence and longevity of Trichogramma may get affected. So a precaution what need to be taken while preparation of Trichogramma, that we need to write the date of parasitation and the species of parasitation need to be noted down on the paper, where card what we are giving for parasitation. So cards, whatever the cards we are releasing in the field, that should be stapled on lower side, so that the tricho card, that eggs of host or the Coursera egg should see the soil. And mostly whether it is parasitoid or predators, we need to release either in the morning hours or late in the evening hours for better adapting to the field conditions. So after releasing the natural enemies, we need to avoid the application of insecticides. If we need the safer insecticide should be sprayed, but mostly we should avoid the application of chemicals. So these are the doses of release of Trichogramma I have mentioned. So it varies from crop to crop and pest to pest. So these are the number of releases to be made and number of eggs to be released per hectare. So this is another parasitoid what we have in laboratory. It is the Telenomus trumus. This is also a egg parasitoid, but Telenomus trumus, it will be mass produced on this Spodoptera litura or Spodoptera frugipeda egg mass. So here one important thing is better parasitation will be there if we give the egg mass within 24 hours. Whatever the fresh egg mass we get, that need to be given for parasitation. So the fresh egg mass we will collect and we will staple on a paper, or we will gum it on a paper, that ovisacs, that egg mass, and we will give it for parasitation. So after 24 hours of parasitation, how we will do for Trichogramma, like that only we will remove that card we have given for parasitation, we will remove and we will keep the fresh one. So the parasitized card will turn to black color within 4 to 5 days. So after parasitation, after 10 days, we will see the adults, the black wasps of Telenomus coming out of the parasitized card. So this is about mass production of Telenomus trumus. Telenomus trumus we will release around 1 lakh per hectare. Mostly the parasitized cards only we can on the lower side of the leaves. So 100 fresh Coursera eggs we will take, and we will not expose it to the UV light. Here we will only the fresh and the Chilonas black bunny adults. So here you can see the Chilonas black bunny adults, the black wasp, and this is the card we have given for the parasitation. So after 24 hours, we will bring this card out and we will keep it in a feeding material what we use for Coursera. In that we will keep this card. So that after 3 to 4 days, the Coursera eggs will hatch out, and that larvae will come out and they will feed. So within that larvae, we will have the Chilonas black bunny immature stages, and that will be developing. And when that larvae will be killed, after 30 days, the adult emergence can be observed in the rearing jars. Here we can see the black wasps of Chilonas black bunny coming out of the parasitized cards. So this is a black wasp, that is a Chilonas black bunny. This is used for management of pink bollworm, and it can be used for management of brinjal fruit and shoot borer. And so 2 releases we will release at a weekly interval, or 3 to 4 releases also can be done based on the need. So mostly 50,000 adults per hectare is the recommended dose, and it can be used for a management of potato tuber moth in godowns. In that case, 2 adults per kg of potatoes is released. So this Chilonas black bunny we will release in the adult stage. So another one is mass production of Gonios nephrantidis. Gonios nephrantidis is a larval parasitoid, that is late larval parasitoid. So here we mass produce on Coursera cephalonica 4th or 15th star larvae. So mostly we will release 10 adults per palm of coconut we will release, and 4 releases we will do. This is the life cycle of Gonios nephrantidis. This is the ant-like adult. What you are seeing in this picture is a Gonios nephrantidis. So after adults of when we observe this, when we have this culture of goniosis, we need to separate the male and female. And male and female we can easily separate with the help of the size. The females are always bigger in size with the broader abdomen, and males are shorter one, and they are short-lived. They will not live more than 2 days. So these are the eggs. What you are seeing is the Gonios nephrantidis eggs laid on the host body, and these are the larvae of Gonios nephrantidis. So when they grow, the larvae will become separated from the host insect, and they form the silken cocoons, and they enter into the pupation. The freshly formed pupae are the white in color, and there will be some color change before the adult emergence. So here you can see the Goniosus adults. You can observe the size difference in the male and female. So males are shorter and females are bigger one. And this is the identification mark. And these are the pupae what we use for the transport. So at this stage, if any farmer or any scientists are required for the culture, means we will transport mostly in the pupal stage. And Rajan, that is a gregarious ectoparasitoid and very useful parasitoid. This is used for management of larval stages in different crop ecosystems. So in this bracon, we have 2 types. One is bracon habiter and another one is bracon brevicornis. So in our NIPHM, we have bracon habiter. So here you can see the female of bracon habiter, which is easily identified with the help of a long ovipositor female. It is laying the eggs on this host insect. This is the larvae of greater wax moth, that is a Galleria melanella. So most of the bracon can lay the eggs either singly or in groups. So once the ovipositor Bracon is inserted into the larval body after insertion of ovipositor. So here you can see the life cycle of Bracon habitat. As I told you, they will lay the spindle-shaped eggs pale color, so either in groups or in singly. So the fresh larvae of this Bracon, when it hatch out, it will be pale pink, baby pink in color, but when it matures, it will be creamish yellow in color. So after 2 to 4 days of larval period of Bracon, it enters into the pupae by forming a silken cocoons. So after a week, pupal period will be a week, and after 7 days of pupal period, the adults will hatch out from the pupae. So this is a gregarious parasitoid. So in the field conditions also, when we observe the larvae attached with the silken cocoons, so that will be mostly belongs to the Braconidae family, parasitized larvae. So one single female can lay up to 2 to 8 eggs, and a female is capable of laying around 229 eggs with an average of 142 to 345 eggs. So female lays an average of 2 to 27 eggs per day. So number of eggs laid will be dependent on the host larvae size. So the bigger larvae can accommodate more number of eggs, and the smaller larvae we will get very few eggs. So a single, so sandwich method means it is simply like a sandwich, the vegetables what we use, that will be sandwiched between two bread. Here the larvae, host larvae will be sandwiched between two muslin cloth. So I will show you the Bracon will be reared in this type of glass jars. So first the adults of this Bracon will be released in the glass jars, and that will be covered with the help of a muslin cloth. And above the muslin cloth, we will keep the late instar stages, that is 4th or 5th instar stage of Coursera. We will place on the top of the first cloth, and to arrest the movement of this larvae, we will cover again one more cloth, and we will tie the cloth with the help of a rubber band. And that larvae will be sandwiched between two cloths, and then the larvae, the females of Bracon from the lower side, that will insert the eggs into the larval body, and that paralyzed larvae we can remove, and we can keep it on a Petri plate for larval emergence of Bracon. So here you can see this first photo, these are the larvae of Bracon, and when it enters into pupation, the larvae will get separated from the host body. Here you can see the dead larvae of Coursera, and these are the pupae of Coursera. So prepare the pupal cards for field release, and we can release either in the, we will release that in the adult stage. So for mass production in the bulk, we can use tub method. It is nothing but the tub what we use for mass production of Coursera, the same tub we can use, or we can take the 4th or 5th instar larvae of Coursera cephalonica, and we can release in the one tub which is provided with feeding material. So two types we can do the mass production. So for initially 250 grams of roughly ground the sorghum, how we will prepare for mass production of Trichogramma, the same way for Coursera, the same way we can take the feeding material in a small tub, and within that we will release 200 to 300 larvae of 4th or 5th instar. So in that we will release the Bracon adults. So that Bracon adults will parasitize the this 4th or 5th instar, and after 10 days we will see the adult emergence of Bracon. So within the tub, we can get around 2,000 to 3,000 adults per tub. So this is a bulk mass production of Bracon. So mostly we will release 3,000 to 5,000 adults per hectare, and adults will be taken in the beaker, and they will be released by tapping the beaker on the crop level. So we have another parasitoid, that is Acirophagus papae. This is a parasitoid of mealybugs. So this parasitoid will prefer only the second nymphal instar of mealybug. So that is Paracoccus marginatus. So each female of this Acirophagus papae, it will lay around 50 eggs in its life cycle. The life period of this Acirophagus will be around 35 to 40 days. So within this period, it will lay around 50 eggs. So mostly the single egg only laid within the one second instar nymph of mealybug, it will lay single egg. So as it is a solitary endoparasitoid, only one adult of this Acirophagus papae will come out from a mummified mealybug nymph. So for that, what we need to have the mass production of this papaya mealybug. So papaya mealybug, either we can mass produced either in the papaya seedlings or cotton seedlings, seedlings or mulberry or growing of papaya mealybug on potato sprouts. So these potato sprouts, when we get good quality of this mealybug colony, we will release this Acirophagus papae into that. And parasitoid of diamondback moth. Cotetia flucella also prefers the second larval instar of diamondback moth. So what we will do first, for maintenance of this culture, we need to have the culture of diamondback moth. If we have the culture of diamondback moth from the field, we can collect the larvae, or we can maintain the DBM culture in a mustard seedlings. So when we have the second larval instar of this DBM, that larval instar we will collect and we will keep it in the cage provided with Cotetia flucellae. So there we need to provide 10% or 50% honey solution as a food for this adult stage. So after the adult emergence of this Cotetia, there will be pre-ovulation period for 24 hours. After that, it will start laying the eggs. So after 24 hours of parasitation, we will remove the larvae of second instar and we will give the fresh group of second instar larvae. So the parasitoids are again provided. It will be repeated until the parasitoid dies. So this is how we will mass produce and repeat the mass production of this Cotetia flucellae. This Cotetia flucellae in field conditions also it is available, and the number of releases, 3 to 4 releases we will do depending on the pest load and the crop stage. And those of release of this Cotetia flucellae is 3,000 per hectare, and mostly we will release the in the adult stage. So here I have given the microscopic picture of this larval body of this parasitoid in the DBM. So these are all about the different parasitoids that can be easily mass produced at laboratory levels, at farmers level. So whatever the parasitoid or predator we mass produce and we release in the field, after release in the field, we need to go for conservation of practices. So as Sir told, we need to have some flowering plants which will be the shelter, and that will be providing the honey or nectar needed for the adult stage. Mostly the adult stage of the parasitoids, they are free living. They will be feeding on nectar and pollen of the flowers. Here in laboratory condition, we will provide the 50% or 10% honey solution as feed, but in general field condition, they will be feeding on the flower nectar. So some parasitoids like aphids, they may require the honeydew for alternative food material. So for that purpose, some places they will be spraying the sugary substances. So that will be acting as an alternative food for the aphid parasitoid. So when there will be shortfall of crop canopy, we can have the short duration crops like alfalfa, brassica family crops or winter crops in between the orchard that will be acting as the overwintering sites for many of the natural enemies, and meanwhile they also provide pollen and nectar. So these are details about the on-farm mass production of different parasitoid. So if you have any queries, you can ask me.
Thank you, madam.
Thank you, sir.
The session is open for discussion. Any participants have any doubt, please, they can discuss it.